Hello and welcome to the Genetics Podcast. I'm your host, Patrick Short. My background is in population genomics and studying the genetic causes of rare disease. I did my PhD at the Sanger Institute in the University of Cambridge and have been in biotech since 2018 when I started Sonogenetics. Sonogenetics helps academic and industry researchers to run large-scale genetic testing programs that speed up their clinical trials, generate data sets for the next big breakthrough, and give participants the best possible experience taking part in research. Each episode of the Genetics Podcast, we bring you insights from the leading minds in genetics and precision medicine, including household names and Nobel Prize winners, as well as early career scientists and biotechs working on the next big breakthrough. Whether you are a scientist, entrepreneur, executive, patient advocate, or simply someone curious about how genetics shapes our world, you're in the right place. Thank you for listening, and let's get started. Welcome, everyone, to the Genetics Podcast. I'm really excited to be here today with Jakob Steinfeldt, who's the co-founder, chief scientific officer at Pheiron, and an honorary research fellow at University College London. We're going to talk today about a really exciting new startup that Jakob has been involved in co-founding, which is a company called Pheiron. They're a small company today, but I think they've got a really exciting and interesting mission that really centers around how do we get a better understanding of why we actually end up developing disease in the first place, and what are the interesting and maybe not so obvious early markers of disease that we can pick up through machine learning and AI and looking at phenotypic data in a lot more detail rather than the really simplistic look we sometimes have around biomarkers that lead to disease. Can we develop a much more accurate and comprehensive picture? And for obvious reasons, this could help us to better predict disease before it happens and treat it, but also to inform drug discovery at the really earliest stages and help people design clinical trials that are a lot more likely to be effective because they have what I think Jakob will describe as more of a GPS type system, a next generation system for understanding how disease is actually caused and developed. So without further ado, Jakob, thank you. I'm looking forward to the discussion today.
Thanks, Patrick, for the introduction, and looking forward to the conversation also.
I would love to just start with Pheiron. What made you decide to start the company, a little bit about what it does, and also your scientific background, your academic background that led you to this. It'd be great if you could talk a little bit about how those things came together and inspired you to start the company.
Yeah, absolutely. So thanks. So what we are doing, basically working in drug discovery, right? So everybody's interested and everybody wants to get their drugs to the market because there's a massive amount of unmet need we have at the moment. What we see is that like 50% of the trials at the moment, they fail because simply the medication is not working. The target oftentimes is not working or has some safety effects which are not making it a viable program and bringing it to the market. And so this we have at the same time on the other side, we really have that massive problem that we have no fundamental understanding of how many diseases develop. And this is really these two things together what has been the reason for us to found Pheiron, because we think they're intrinsically connected. They're intrinsically connected on like the most fundamental core questions we face. They're connected in the part of, do we choose the right target to treat the disease? Do we have the right markers to identify the right people to guide the clinical development? And do we get the populations right to treat the trials in, and therefore have the pivotal results we need to bring them to the market? And so this is sort of the vision we have, like using that understanding, that phenotyping to build the GPS, that map that gives us the evidence to really help de-risk on the targets, on the biomarkers, and on the population. So that's about the company. And there we work together with partners. And the question was in terms of what made us found the company in the first place and how did we get there? And I've co-founded the company with my co-founder, Tora Burgel. And so we both met at Charité. So I was doing my postdoc at Charité. Tora was doing his PhD at Charité. And we worked at risk modeling. And for me, risk modeling came from, it was not a direct path. So I actually started out wanting to come in, like loving to play with computers as a child, but then sort of ended up really liking more being with people and went to study medicine and really fell in love with the human interaction, the human part of medicine. So for a long time thought really doubling in there. And so we did like the standard like wet lab many people do, right? You try many things and fell absolutely in love with the computational part of that science. So I did my wet lab studies there in really deep alternative splicing. But what I got really interested in is that cell phenotyping part of things. So like at the time, some of you might be familiar with it, like multiplex stainings. There was like Ann Carpenter cell atlas, super cool things where you basically intervene on cells. You have multiple multiplex readouts to characterize them really, really well. And this is really how I got the connection from the computational into the medicine and the ML. And at the ML part, I mean, at the time, it was a few years after AlexNet when all this deep learning really took off. And so I was super excited about the potential we had there, but also all the time had my clinical rotations and my clinical work where I realized, look, actually, we have such a problem connecting all this information in the first place. And it's just like all this theoretical potential, but the reality is so much more complicated. Was a bit in between sort of I always sort of liked cardiology and wanted to start sort of on the interconnection of cardiology and science. I came to Berlin to Charité and was working my first postdoc sort of combination, and it's now the German Heart Center of Charité, on well, how can we build a risk models to identify individuals who will get cardiovascular events. So that's quite it's extremely important question, but as a narrow question. And so worked there on how we can possibly introduce polygenic scores to identify some individuals before they really get clinical symptoms. And that is sort of in that part. And then the second postdoc working with Dr. Langbeck and sort of a bit more the prognosis part and getting also more into genetics, realizing that the actual fundamental question is that we don't know for most disease we have out there who gets disease in the first place, who gets complications. And this really has been the foundation for us in the combination with that massive problem that most programs fail, and realizing that genetic support is a core aspect in that equation, a core factor to the risk. Right biomarkers are core factors to the risk, and making the trial effective are core factors to the risk, and they're all inherently linked to phenotyping. And this is why we are so convicted in building the GPS for the map to really now help people navigate these three issues, like the right target, the biomarker, and the population.
Yeah, maybe you can talk about heart failure or cardiology more broadly as an example of the gap between where we need to be and where we are today, because I think you made the point that there's we have very crude measurements of risk and progression today, and we know that they don't capture anything near what an archetypical GPS that could tell you exactly when you're going to get there, how you're going to get there, what the path is you're going to take. I really like that metaphor for that reason. But what's missing, and to what extent do we not know what is missing? And we've got to figure that out. Is it that actually we have all the biomarkers, but the difficulty is getting them together and making sense of it, or is it actually there's some fundamental discovery work to do?
Yeah, thanks. So there are many. It's a complex question, right? But maybe let me try taking typical disease progression as an example of what is the problem at the moment. So I shortly touched upon the first part already, the primary prevention. So primary prevention of cardiovascular disease is a massive problem because at the end of the day, people don't feel they have a problem or a sort of catastrophe building up. And at some point it's like bam, and they have a massive problem where they nowadays typically don't die initially anymore, but they basically lose, heart muscle dies, they get into heart failure. And that's like a horrible disease with like a massive burden on their everyday life. So we, at the moment, have a few markers which are established to identify who will be at high risk if they don't have symptoms. So, the most sort of fundamental and strong one is a very silly one, is age, right? Like the older we get, the sort of more risk we have. But then there are like others established, like some of them as just a few examples, like the cholesterol we have, in particular the LDL cholesterol, some hypertension we have, some comorbidities we have, like several of these factors, blood pressure, where we have like the typical scores, like many people of you might know, the score, a CVD score, the Q-risk score, which try to catch up with some of them. And it's really difficult to implement them at scale, and they're not actually they're much worse than we would like them to be. So we still have a ton of people coming in with a heart attack, and we simply didn't get them on the right prevention treatment before. I don't want to talk so much about sort of the clinical part of that because this really is here in this context also incredibly important for the development of these drugs. And so going on, they come in and then they have a heart failure. In that case, it's probably heart failure. Most likely case is because ischemia. They have the artery clogged. But there are a ton of other things out there. Like the signpost is they come in and they have chest pain, they have dyspnea, and then they have a few measurements, drop in intake, and they have an ECG-like changes, and then they have a coronary intervention. But there's a ton of other pathways there. Just one example, ATTR amyloidosis. Like you have very similar symptoms. Like it's very easy to confuse it with a heart failure phenotype. And still, like there's a massive burden. And we don't get it right it's extremely difficult to get it right with the signpost and this system we have there in place at the moment. And so this is our core conviction why we think we need that map and we need to build that GPS to get that right initially in a context where we use it for the discovery of the targets, for the biomarkers, and getting the population right. And prospectively, I mean, it's a fundamental concept, right? We need a systematic, quantifiable basis for disease. And your question was, what is actually the problem? Do we have everything there, and we just need to analyze it? I mean, the problem is on multiple fronts, right? So the first one is we fundamentally have only in the last 20, 30 years the possibility to actually have information on so many people like 100, 10, 100,000, up to million people in the population measured. So it's the first time we actually can do that. And that's a requirement. And then only in the last years, for many of these particular broader assays, give us a glimpse on where the world could be, like come available. So I'm thinking on, like we worked on some NMR metabolomics. You can think of a lot of interesting work done and with more broad metabolomics, or also the newest assays, I mean, basically advertising, basically offering up to like 11,000 proteins measured at a time. So this is really, the more complex these assays become, the more we cannot manually like analyze every individual part, but our conviction is that we need to use the tools we have at hand, and they are data-driven to answer that simple question, who gets disease, who goes on having complications, to, again, come back to these three things, like get the target right, get the mark right, and get the population right.
Yeah, and I think something interesting that you said there's an obvious application of predicting disease early, which is in the healthcare system. We'd all like to know a lot earlier and we can make changes and interventions. But actually there's, at least I think to most people, a less obvious route, which is the problem that you can solve in clinical trials with this, which is in heart failure in particular, you have to run trials for many years with many thousands of people for the reasons that you can't predict which of them are going to get heart failure. So you enroll based on some very broad risk factors, age being one of them, but some of these other biomarkers, but then you're waiting for years in some cases for people to actually have the event, where it sounds like with this kind of approach, you could run clinical trials a lot more quickly, because if you know with much more specificity who's actually much more likely to go on and have an event, then you enroll fewer people, you put less people on an experimental drug that don't need to be because they're not going to have an event in that time. So maybe you could talk a little bit about that problem space and what we might be able to do there, because it seems like it's a little bit of a hidden bottleneck here. People don't really realize how many drugs maybe never even get developed in the first place because someone looks at a budget and says this clinical trial is going to cost me $100 million because it's going to take 5 years. And if it could cost half that, then actually maybe we would do it.
Yeah, thanks, Patrick. So the underlying thing there is, if you develop a new drug, the core thing you need to do in the first place is you need to sort of de-risk your investment, right? Otherwise, you will not be able to sort of get the thing going, or it will be a oftentimes bad investment. So what this means is the initial trials, and if we know that 50%, 60% fail because the drug doesn't work, needs to be as cheap and as fast to prove that the drug itself has a chance to work. And that is a massive pressure driver. So, and as you mentioned, it's extremely complicated if I don't know exactly who in the population will get on, let's say, prevent the progression of heart failure. And that's exactly basically the problem we have. So to do that, to identify, we have basically two core levers at play here. The one core lever is this established concept of prognostic enrichment, is making sure that you include the people who will actually get the events. Another concept is something basically surrogate endpoint. Is quite a common used term. If you can identify something we can measure, which will give us the answer whether the drug works, yes or no, earlier than waiting for many years until a hard endpoint is met, that's an option. And the core problem with these things is not that the principle is not clear, but it is that you oftentimes simply don't have the means to know who will get in or what to measure. And this is really where our core conviction, again, is the phenotyping comes in, and where we see that based on these population cohorts and based on the work we did and we're doing at the moment, you really can use that to filter out and stratify the population to tell, look, these people are going to have 10 times or 20 times as many events as others. Therefore, include them in the early trials. So I can basically be very specific and much more specific here. How can we do that? We can do that, of course, by looking at the age, which is oftentimes a good starting point. But there are so many things we can measure nowadays, and they go from the very basic things in, let's say, heart failure trial. Most of these patients anyway will access to some basic blood measures like the cholesterol levels. We will have many of these settings ECGs available. So we know that in ECG there's a ton of hidden information we can simply extract and use to tell who is going to get these events more likely in others. And there is all of this amount of different data sources out there. They are like echoes. They're like, depending on the trial, we can even consider MRIs, but there are also specific proteins you can use to figure out who's going to be at a high risk. And what we see is that this map is not there at the moment. And this is really our core conviction, that with that map, that can really give us the entity we need to navigate. This is what you have to measure in these people to get the biggest likelihood that you have the right people in your trial. And therefore, the trial will either faster get you to the answer, is the drug likely to work or not? Or you simply will have to include much less people. And there is some this phenomenal thinking about it. Some of these like you can simulate it with you don't need so much predictive information to really massively reduce the size of your trial. And as long as the screening is somewhat scalable and cheap, there is so much potential and it's not used at the moment. So this is really where's a core value for the platform.
Yeah. I think it's a really great idea, and I've heard this in the context of cardiometabolic disease, for example. Perhaps you use something like a polygenic risk score, or polygenic risk score in combination with some of the more conventional scores. Obviously, there's no point in just using it by itself. You could think about some of the new technologies like proteomics. If maybe we took an example of using polygenic risk scores, for example, to identify the top decile of people who are going to be much more likely to have an event in the time horizon of the trial. One of the things I've always wondered about is let's say that phase 2 trial is successful and you're actually able to show that there's an effect on the primary endpoint in that subset of the population. What would someone do for the phase 3? Would they go after that small subset of the population again, or would they broaden out to the whole population and basically use the phase 2 as a little bit of a proof that the drug works in at least some subset of the population? Maybe you could talk me through how, like, a biotech company, because it seems like something that a pharma or biotech company would want to do to try to get that signal quickly in a phase 2 and figure out, do I have something here? But then it maybe complicates the decision in the phase 3, right? Because you haven't tested it in as big of a population as possible.
Yeah. I mean, there's an inherent trade-off, right? Like, if you reduce the amount of the population viable for that trial, you reduce potential market later. But what we the things we heard and a lot of times here in these conversations is the core question they want to make sure with as minimal investment as possible to really focus on their core bets. So they want to try many bets, and if they don't see the signals, they simply will not go on. And I mean, if they then on that basis go on, I mean, what if you have a good signal, that gives you a massive amount of confidence because you have already this core factor de-risked, that number one can help you to get the sample size right as you would need, and therefore estimate what it actually would cost to get to that, to cross that bar. Or say, look, I mean, I might have to work together with a very big pharma company to finance that trial, or basically refocus their bet on the 3 other assays or programs they might have, where they see they need to be much less stratification to get to clinical relevant effect. But our core conversations we have are around the risk everybody has on their mind. How can we make sure that the things are actually working as early as possible? And this is really the core question we perceive and we where we see the core value. And maybe let me add like one thing there. Like you mentioned the example of the polygenic scores. I think it's an interesting example because everybody talks about polygenic scores, and I mean they're sort of really, really cool in the sense that once you have taken the genetic material, they're quite broadly accessible. You can use them for many things. But the interesting thing is that that's not just the case for like genetics. That's the case for actually technically the case for anything we measure. And practically we know that many things we measure are relevant for diseases, not just for quite a broad set of diseases. So for example, like take the example of an ECG. An ECG is, I mean, many diseases have cardiac phenotypes. Like I mentioned the ATTR amyloidosis before. Like there's sort of many sort of in particular immunological, rheumatological diseases which have phenotypes of the heart. And there at the same time, many diseases which are mostly sort of confined to specific measures because traditionally they're treated by certain specialties. So it's a very interesting thing. And what we know and what we found is that many of these data sources are really informative for diseases across specialties. And that's an opportunity to, number one, pinpoint which one for which disease, and then how can we scalably implement that in these decisions towards the target, towards the biomarker, and towards the population.
Yeah, that's a very interesting insight. So what does that imply about what kind of data you need to build this GPS? Does it push you towards wanting more larger and comprehensive population scale data sets like the UK Biobank, where you've got cross-section of the population and they're just layering on more and more new interesting assays every year, right? They have NMR metabolomics, they have proteomics, they have imaging in a subset. But I guess there's also an argument to say we need to go deep in some of these less well-represented populations. So how do you think about that? You know, is it broad or is it narrow into a heart failure population and you capture much maybe more relevant phenotypes for that population?
Yeah. The thing is, at the end, in the best case, you would have everything, right? But the practicals, you don't. So oftentimes, like how we see it is, you need these population-based cohorts, and they're an amazing opportunity as a basis, to really make hypotheses and get an understanding of what matters for what. And that again sounds somehow silly, but we don't know that for the majority of diseases. We have no understanding how really the risk of sort of how well we can predict the risk to have, let me make out an example, rheumatological outcome based on ECG. I mean, we have some links established here and there, like these signposts, but we have no fundamental data-driven understanding. And going from there is totally clear. Like we need, in particular, prospectively enriched cohorts where we have people who have diverse phenotypes of individual diseases. And I mean, there are a few sort of different ways around that. I mean, one is, I guess, our future health is a good example, sample size. But at the end of the day, what we think the direction needs to go is like this massive health systems which routinely take care of people with very diverse specialty phenotypes. They measure a lot of diverse assays and they have associated biobanking. So you can really get the routine care interlinked with the research and can really trace back and go back, look, these are now the specific populations I want to specifically follow up and go in. But that's more of a maturation, iteration, and where we want to go. And at the moment, using like populations and using like data, also not particularly enriched, is an impressive opportunity, an amazing opportunity, and we're super excited about it.
Yeah, what's the bottleneck there? Is there a shortage of kind of health systems that have very robust research arms and want to do these kind of collaborations, or are they too expensive? Because I'm thinking like this is an interesting challenge in that most health systems in the US and in the world are collecting this kind of data at scale, but getting access to it is probably the most difficult part. I'm wondering how much you've tried to get access to some of these kind of populations that you described and what challenges you run into.
Yeah. Yes, the answer is of course. I mean, we are basically actively looking for partners there. And the thing is that what we found is that many of these health systems are extremely interested of making unlocking sort of the data they have there. And the phenotyping is, of course, a core component of it, because the images or the assays oftentimes, they don't tell you much, or at least not in a scalable way. So what you need to do is nowadays, you have this massive opportunity with data-driven approaches to unlock them. But it has many complications, because what it means is that you need, in these health systems, a very, very strong expertise to build up these resources. And there's so many core challenges from the way of how this data is basically acquired in the first place, how a blood pressure measurement in an ER gets in a system in the first place, then how to basically link that to somehow standardized representation of sort of these signposts in the first place. And they're like nowadays with SNOMED and many of the vocabularies, there are sort of the semantic foundations for many of these things. But to get the right people there in the first place that build up systems that alone on a technical level sort of work to the extent is something many, many health systems struggle with a lot. And so this is the first basically barrier. And the second one then is, of course, there are some who clearly identified the value, and then, they work together with companies, and I guess with the obvious ones, we also don't want to go too much into the names now, but we also have conversations or already work together.
Yeah, it is. I think the way you described it makes sense in that it's just outside of their standard course of business, right? They're first and foremost trying to deliver care. And so to spin up a best-in-class research arm requires all the skills and expertise and time and things like that to clean the data and make sure that it's fit for that alternate purpose, which isn't really their primary. And so that's where I think big programs like the UK Biobank have just found a way through that. Yeah, well, it's not really even through the middle, because they're not doing anything from a clinical perspective. They're kind of really started on the basic research, and then have, I think over time, they'll become more and more clinically relevant what they do. But I'm always interested in the organizations or institutions that manage to thread that needle between the two. Like Genomics England, I think in the UK has done an interesting job of being both healthcare system service first and foremost, but also doing a really robust job on making data available to researchers. But it's much more common that people just focus on one or the other, right? And don't straddle that line very effectively.
But at the end of the day, that feels like that is really one of the core opportunities, like moving into the next decade or decades, right? Because I don't remember exactly where this comes from. I still remember, like many years back in my studies, there was I studied in Germany, right? So like there's a lot of conservatism around data sharing, and I think it was from Estonia, like a representative who was there on a conference. He said that at the end of the day, every patient who comes in the door feels the privilege of that, basically that the care they get is based on the experience of all the other people who had received the care before, and so there's a strong sort of shared commitment to then also share back the data and the experiences learned from their case. And the interesting thing is, in most cases, is exactly the people who are most at need who are the most open to sharing this. And this is really where we see an amazing opportunity to fundamentally sort of increase and improve the possibilities we have. Absolutely in the clinical, but also absolutely in the development and drug development, from the initial target to the market and the population at the end.
On a more personal level, how did you make the decision about starting a company versus continuing with academic research, medical pursuits? Because I always ask this question because I made that decision myself about 6 years ago now to start a company instead of going into academia. But I think it's a very challenging decision that a lot of people come to in the end of a PhD or end of a postdoc or in the middle of the postdoc and try to decide if they're going to leave, perhaps in some cases temporarily, but leave that world where they've spent so much time building a reputation, really going deep into a particular area. And then when you go and start a business, some of the skills are transferable, but then there are many other things that you spend years on that would not be rewarded in an academic context. I'm just wondering, how was it difficult or easy to make that decision of actually starting the company in the first place when you did?
I think at the end of the day, it was an easy decision because I realized quite fast that I'm in love with science, and I really got hooked on that question. Sort of how can we improve the understanding of who gets disease, and at the end of the day, use that to make an impact. And I know that there are many paths in academia, and I had really great sort of also support there I could have gone and I could have been also happy with. But the opportunity to really build a team laser focused on the mission to really move the needle and see the impact of what you're doing with everybody aligned on that with the right incentives is something where I'm convicted right now that I know that for the question and the ambition I had, that was the right decision to make in the moment. And I'm extremely excited sort of about the opportunities ahead. Yeah, so it was really in that moment the opportunity I saw. I simply needed to give it a go. And to be fair, also like together as my co-founder, I think we had a really cool constellation where we both were really excited on going that, and now really see so much need that every day sort of it feels reinforced.
That's a good feeling. And what have you learned? Like what have you had to learn, especially skills wise, that's been different from what you built in your PhD and postdoc?
I mean, I've never built a company before, so, and neither has my co-founder, at least in the part. So there is, I think the core learning has been that there's so much things going on that in every morning you have to ask yourself, okay, what individual thing brings your company or brings you closer to where you want to go? So constantly aligning on where you want to go, and then every day leave all the rest there and really making the decision. And that is sort of hard sometimes because everything is sort of important, but it's also extremely gratifying because you look back then a month and 2 months and 3 months and you really see there's a nuts amount of things you can achieve if you are excited about something. You feel there's value in it and you have the right setting and can also sort of decide on where you set your priorities to a quite large extent. Yeah, so this is really, the progress is immense there. And so this is certainly something and we had to learn, but it's something which is extremely exciting because it moves extremely fast.
Yeah, definitely. And what are you most focused on right now? Like we've talked a little bit about the, I think, broad vision of what you're looking for, but in the next year, next 2 years, what are the kinds of things that you're most excited about? And feel free to make call outs if there are particular kinds of partners or other companies that you want to work with or that sort of thing to help you get there. If you'd say, I really need, I'm interested in accessing partnerships or data sets in this particular disease area, or we'd like to help companies that are working in trials like this. I'd love to hear a little bit about what, in the next year or two is going to make you really excited.
Yeah. So I think I would say 3 things. The first thing is that we see that there is this massive need in particular sort of in also smaller biotechs to get that guidance on integrating this evidence from the populations into their programs. We see there's this massive need every day. So I'm extremely excited working together, partnering with these biotechs to help them using the best possible evidence for their programs. And this is something I sort of really learned to sort of really enjoy over the last month because it's a massive confirmation that we can provide value here. I think the second part I'm personally really sort of excited and looking forward to is, as we've talked about, I mean, data is really a core part of what we do. And we have like a ton of really, really exciting ideas, which we plan on materializing over the next months on moving some of the more unconventional ways of really getting the right data for that phenotyping case, like making sure we have the right enriched populations, making sure we have the right assays measured in the right diverse populations. So this is something I'm really excited about. And the third part is, I think more of an excitement sort of from a scientific technical point of view is like, I love sort of building things and notice how they get faster and better and mature and then seeing what comes out. And so, I mean, this is something which natural over the next 1-2 years. I know in 2 years, it will look very different to how it looks now. And I'm extremely excited how it will look. And yes.
Yeah, one of the things that's always stuck with me, I think this is the case with most things in life, but certainly with research and certainly with starting a company, is you often overestimate what you can do in a month, but you underestimate what you can do in a year. And so it's really kind of dramatic sometimes to look a year back and realize how much you've managed to do. If you're looking at a week or you're looking at a month, often it doesn't always feel like we've made a massive improvement because sometimes, the improvements, they come in fits and starts and they're not always all happening at once. But I definitely share that piece with you that if you look back over certainly a year or two, then you'll be amazed at actually how much you've accomplished in that time. As long as you're doing that thing you described every day where you're picking the one or two most important things and you're really focusing on them and you're coming back every morning to say, like, this is my focus for today and I'm not going to get distracted.
Absolutely.
Well, thank you for doing this. This was a ton of fun. I learned a lot. If people want to keep track of you, if they want to learn more about the company, where should they find you?
They can find me on Twitter, on LinkedIn, or they can just drop us an email. They can find us on our website, www.pheiron.com. And just any of these channels. We're very excited to hear. And I mean, think specific things or explore synergies and any other point people might be interested in.
Wonderful. We'll put the link to the website in the show notes so people can find you easily. Thank you so much. I really appreciate you taking the time and joining us. It was a lot of fun.
Thank you so much for the conversation.
That wraps up this week's episode of the Genetics Podcast. I'd like to give a huge thank you to our guests for sharing their valuable insights and experience. And thank you as well to our listeners, as always, for tuning in. If you enjoyed today's talk, the number one thing I would really appreciate from you is if you could share it with a friend or colleague who you think would enjoy it as well. We would also really appreciate if you could subscribe to our show and give us a quick rate and review on Apple Podcasts, Spotify, or your favorite podcast platform. Both of these things help us become more visible when people search for genetics and precision medicine podcasts. And we're always eager to hear from you. Please reach out to us with any questions or feedback on social media. You can find Cynogenetics on Twitter, LinkedIn, or visit cynogenetics.com. Finally, a big thank you to the team behind the scenes who make all this possible. In particular, Amy Cousins and Sonia Shah, who produced the show, and James Pierce from Selected Frequencies, who handles the audio engineering. I'm Patrick Short, your host, and it's been a pleasure. Thank you, and we'll see you next time on the Genetics Podcast.